not going to do it. We're just going to...
Hi, this is Miss Litton, and this is lunchtime. Say hi.
Hi.
And this is AP Biology, and we are reviewing meiosis and sexual reproduction. Okay, so the first thing we need to get down is the whole idea of haploid versus diploid, an N and 2N. And I want to make sure that you feel comfortable with that. And the idea of alternating between, we make haploid cells like egg and sperm, and those are the only haploid cells that you and I make. And when egg and sperm join together, they form a diploid zygote. Once you have a diploid zygote, it will only undergo mitosis until you get ready again in our ovaries and our testes to do meiosis and get down to half the number of chromosomes. Comparing mitosis to meiosis, meiosis has how many divisions? Meiosis has how many divisions? 2. And in meiosis 1, what are we separating?
No, the homologous pairs.
Homologous pairs, the mom-dad chromosomes, right? And in meiosis 2, we are separating the sister chromatids. Which one, meiosis 1 or meiosis 2, looks most like mitosis? Which one? Which one looks most like mitosis? Meiosis 2, exactly. Why? Separating sister chromatids, which is what we do in mitosis. Do you see that? Okay, so we'll talk about some deviations with plant cells here in just a minute. But what I want to ask you is, if I said, I need a new page. If I said the diploid number was 16, how many chromosomes would I find in a sperm or an egg? Think it out in your head. If I said the diploid number was 16, how many chromosomes would I find in a sperm or an egg? All right, how would I write diploid number is 16? How could I write that? That you would understand it. Yes?
16.
Okay, but was 16 equals what? What equals 16?
16 equals 2N.
2N, that's exactly what I wanted. Okay, so 2N equals 16. How many chromosomes would I find in a sperm or an egg? 8. And how would I write that?
8 equals N.
Okay, good. How many homologous pairs did this cell have? How many homologous pairs did this cell have?
4.
Yeah, it doesn't matter if you say 2N or N. I could give you the N number of 8. I could give you the 2N number of 16. But what I'm asking you is, how many homologous pairs does this organism have? 8. It has 8 homologous pairs. Okay? 8 homologous pairs, and it doesn't matter if I'm talking about the gamete or if I'm talking about a somatic cell. That part stays the same. For us, how many homologous pairs do we have? 23. And it doesn't matter if we're talking about one of our body cells or one of our gametes. For us, the number of homologous pairs we have is 23. Do you see how I'm differentiating that? Any questions? Being quiet and eating your pizza. Did y'all have pizza yesterday and you're bringing in book? Okay. That pizza was so good. I need to know that pizza that you got that. It wasn't as delicious. You're talking about the chicken barbecue one, right? Or not barbecue. What was it? Chicken pesto? Chicken garlic. Oh my gosh, it was so good. Okay, anyway. Period 3. That was awesome. All right. So when you watch a person grow, what are you doing that by when you watch a person grow? Mitosis or meiosis?
Mitosis.
Mitosis. But if you're watching them generate sperm or egg, then you're talking about meiosis. Okay? We want to remember that homologous pairs are the same size, the same shape, and code for the same characteristics. And when you talk about separating these homologous pairs from each other, you're talking about the process of meiosis 1. When you're talking about separating sister chromatids from each other, you're talking about meiosis what? 2. Okay, good. Just so long as you got that down. Now, there are key events that occur at different stages. And what I want you to see, if you can remember, I know you're doing some group shared notes right now, but we have meiosis. Here, let me think of another way to do it here. We have meiosis 1 and we have meiosis 2. All right. And what are our stages that we're looking at? What are the stages? Prophase, then what? Metaphase and anaphase and telophase. So we could be talking about prophase 1 in here and prophase 2 over here, right? Key events that happen in each of these. How would you fill in that box? How do you think I would describe it for you on a test? Maybe insert a table and start filling it in and seeing if you can remember. And I'm going to pause for just a moment. Okay, so hopefully you took some time to fill in your boxes. Let's talk about some key events in meiosis 1 that occur in prophase. Go ahead, tell me. Synapsis, good. So that's when you're forming a tetrad. And what can happen as a result of that? Crossing over. And what's good about that? Yeah, variations. Also, what else happens?
The nuclear envelope.
Yeah, the nuclear envelope and what else? Nucleolus are what? They go away, and then we start to see the what? Chromosomes become visible. That part stays the same, right? Okay, give me key events in metaphase 1.
Homologous pairs line up.
Yeah, okay, so the homologous pairs in the middle, but what's significant about that? They can what? They can line up differently. What do we call that? We learned it when we started. I don't know. Independent assortment. Remember that?
I think I know.
How they line up in the middle. Okay, what happens during anaphase 1?
Homologous pairs separate.
Okay.
Thank you.
And now this cell is considered what? Haploid, because you don't have your homologous pair anymore. Okay, so in telophase you have 2 haploid cells, yes? And then when you do meiosis, it's actually happening. Meiosis 2 is happening in 2 cells, right? Are you with me on that? You don't have to do it like that. I'm just saying times 2 because it's happening in 2 cells. But there are certain parts that are going to stay the same, right? The chromosomes are still visible, but there's not going to be any crossing over, right? No crossing over. Why?
Because there's no homologous pair.
No homologous pairs. Good job. Okay. What happens in metaphase 2? What happens in metaphase 2?
The chromosomes line up.
Chromosomes are in the middle. Can you have independent assortment at this time? No. Independent assortment is referring to when you form that tetrad, your chiasmata, where things can cross over. You don't have your homologous pair. You're single file in the middle, right? Okay, what happens in anaphase 2? The sisters separate. Good. And in telophase 2, what are you going to form? 4 haploid cells. Okay. You good on that? Yes. 2 for each. Yes.
So the haploid cells are the daughter cells?
Yes.
Okay.
What else? We good? Okay. So next thing I want to ask you is, where in this process can we get variety? Where is there a lot of variety that you see? There's 3 big areas. Where? Crossing over is one place where you get variety. Where's another place you get variety? Independent assortment. Where's the third way due to meiosis that you get variety but we don't see it? Thank you. Where's the third way you get variety?
Fertilization.
Fertilization. You don't know what sperm is going to hook up with what egg. So those are your 3 big areas where that meiosis lends itself to variety. Good, good, and good. We did all of that. Let's do these. I'm going to go like this, and you're going to tell me the answer. Okay? Stage of meiosis where sister chromatids separate.
Anaphase 2.
Anaphase 2, good. Name the stage of meiosis where a nuclear membrane rebuilds for the first time. That would be what? Telophase 1, good. Name the stage of meiosis where crossing over occurs. Prophase 1, muy bueno. Now I sound like Trump. Name the stage of meiosis. Let's go, hombres.
Telophase 2.
Where 4 haploid cells are formed. Where?
Telophase 2.
Telophase 2. Name the stage of meiosis where single chromosomes are being formed. Nuclear envelope may be breaking down again. Prophase 2. Okay, name the stage of meiosis where homologous pairs are separating.
Anaphase 1.
Anaphase 1. Name the stage of meiosis where homologous pairs are in the middle. Metaphase 1. Are these easy peasy lemon?
Yes.
Yes. Okay. Name the stage of meiosis where single chromosomes are in the middle.
Anaphase 2.
2.
Yeah.
Single chromosomes. Metaphase 2 is going to look like this, right? Whereas in 2 different cells, it would look like this. Whereas in metaphase 1, it would look like this. Remember, metaphase 1, you're in 1 cell. Metaphase 2, you are in 2 cells. Okay. All right, we did that. We did that. Okay, then we talked about life cycles. Okay. Do we do alternation of generations? Do we as humans, animals? No. Who does that? Plants. Fungi do that as well. Okay. So they have 2 generations. They have the gametified generation and they have the sporified generation. The sporified generation is always 2N. It's always going to make spores by meiosis. Then these spores are going to do mitosis in order to form this gametified generation. In this case, it's a green heart-shaped structure. It is independent of the sporified. And then it will do mitosis to form its gametes, which get together through fertilization, and then we have a 2N zygote again. Fungi do that as well, alternation of generations, but they spend the majority of their time as a haploid. We form sperm and egg. Spermatogenesis, you always make how many sperm? Every meiotic event, you will make 1, 2, 3, 4 sperm. But in oogenesis, eggs, you will only make 1 and 2 or 3 polar bodies. The chromosome number is the same, okay? But we're concentrating our what? Cytoplasm and our nutrition in 1 large cell, okay? Because we're packing the picnic lunch for, our little fallopian journey. All right, so then we want to talk about when things go wrong. So it can be wrong in number, or it can be wrong in structure. Can you write down what are the big wrong number generalizations? And then see if you can think of some structural problems. See what you can come up with. Problems with number, problems with structure. See what you can come up with.
That'd be cool.
You ready? So having your exact right chromosome numbers, you are okay. Euploidy is being just right. If you have another whole set of chromosomes, you're a polyploid. If you're off by 1, you're an aneuploidy. If you have an extra one, that's called a trisomy. If you're down 1, that's called a monosomy. Okay, so this is -1 and this is +1. That's when you're off by 1. Okay? Do you know what the most common trisomy is of all? Down syndrome, yeah, Down syndrome. Okay, and so you can get a trisomy or a monosomy by the term you want to know is non-disjunction, and that's failure to separate. So anaphase is screwing up. So you can either screw up anaphase 1 or you can screw up anaphase 2. Either way, some are going to have extra and some are not going to have enough. Okay? And you can see that when you look at a picture of chromosomes. Karyotype is a picture of your chromosomes. And you can line up your homologous pairs and you can count them. And this individual, you can see all the different homologous pairs lining up, and we can tell it's a boy because they're XY. But we could see in this one there was a difference because here are 3 chromosome 21s. So that would be evidence of what? Not evidence of crossing over, but evidence of non-disjunction. Evidence of non-disjunction. I don't know what you're saying back there. I couldn't hear it. Oh, were you saying Down syndrome?
Yeah.
Yes, specifically Down syndrome, yes. Evidence of non-disjunction. Okay, you can also have the wrong numbers when you're dealing with sex chromosomes. And on those, kind of the big ones that you will see is you will see where the female only has 1 X, and that's called Turner's, and I put that in your notes. Or a male has an extra X, and that's called Klinefelter's. You can also see X, Y, Y, and I think I gave you poly X as well, and there's some other things associated with this, but I would be familiar with Turner's and Klinefelter's. Now, if I'm looking at a nucleus of a cell, could I tell just looking at the nucleus if something was male or female?
Yeah.
Yes, what would I look for? But I can't see them when I see these chromosomes like this, we have to be getting ready to do mitosis or meiosis in order for me to visually see chromosomes. Let's say it's not doing mitosis or meiosis. Would there be a way for me to tell if there was a problem with the sex chromosomes? Where are you not old enough to hang out yet?
Bars.
Bars. I could be looking for bar bodies. Okay? Bar bodies. Bar bodies are your ladies were a mosaic because either one of our 2 X's will condense into a bar body in our nucleus. If we don't have that second X, it's an issue. We have Turner syndrome. But it's like we have too much Xness. If we have both of our X's there, they're large chromosomes. The Y chromosome is very tiny. Holandric traits are found on that. But one of our X's will condense, and so you can see it in our nucleus. So if a female does not have a bar body, then you would assume she has what? Turner syndrome. She only has a single X, so she doesn't have an extra one to condense. If a male has a bar body, you would assume he has Klinefelter's because he has an extra X, so one of those would condense. And we're going to learn about this later. Calico cats, when you see calico cats with their mottled colors, that's because their coloring is on their X chromosome. So it depends on which chromosome condenses, which affects the color of the cat. So calico cats support this, can only be female. When you see that different coloring, that can only be a female. Why is that? It's too much thinking. Mrs. Litton, don't make me think. Okay, so calico cats are mottled colors, and only the female appears mottled. And I told you their coloring is on their X chromosomes. The males do not appear mottled; they're just a solid color. Why is that?
Because
They only have 1 X chromosome, and they display that 1 color, right? But females have 2 X's, so depending on which X chromosome is active will depend what color that area is. Do you get that now? We're going to go over that in genetics in our next chapter here.
That's not going to go over on the quiz, that'd be real.
Okay, so we talked about Down syndrome. We talked about some abnormalities. Here's Turner, Klinefelter's. You can see Klinefelter's extra X. Here we're missing an X. Poly-X, Jacob syndrome with your extra Y. Last but not least, let's talk about a few other things. A deletion. That does sound like a loss, right? So somehow during crossing over, you lost a chunk, in this case the A. And if that A gets added on, did it do that here? Oh no, but let's say this A gets onto this one, that would be called a duplication. So a deletion and a duplication. Now those usually come hand in hand. This is an inversion. Somehow in the crossing over, this one thing got flipped. Okay, that would be an inversion. And a translocation is when you cross over not with your homologous pair, but with some other number. So let's say chromosome 8 and chromosome 13 get together and exchange information and do crossing over. That's bad, and that's called translocation. All right. Do you know when that one is?
Well,
What one do you think? Yes, Klinefelters. Well done.
okay. That's cool. Are these gonna be on the quiz?
These are pretty hard. This one you should get. That one was a real
Let's
specific
not
one.
talk for a second.
That's translocation.
Exactly,
Did
that
you
is
know?
translocation. Because she saw non-homologous pairs and they were exchanging. That's translocation. I'm in the wrong location. I translocated. You should know the answer to this. A, Down syndrome.
I was pointing to the board. Oh,
Turner's. It's Turner's syndrome. Inversion. Good job. It's an inversion because it's flipped.
the only one I know.
Poly-X.
The one you kept saying like the entire lesson.
Poly-X. You were saying it again and again?
Yes. Yeah.
Yep. Deletion or duplication. Good.
You guys, let me know if it's in the name. Jacob syndrome.
Jacob syndrome.
Because they go to prison.
Yeah.
Jacob.
The end. Yay!
Yes.
Good job. Are you glad you stuck around?
Yes.
Pretty
All right.
much.
Don't stay up too late, and if it's super late, eat a piece of
Those.
toast. That's what I was saying. And go to bed. Go to bed. Get some sleep.